Well, thank you very much for giving me the occasion of presenting my research project, and thank you for the grant that will support this research project continuation. There are different types of mutation which are responsible for Duchenne muscular dystrophy. About 30% of are point mutation, that is a change of only a few nucleotides. And although there are thousands of nucleotides in the human genome, the change of only one nucleotide can sometimes lead to an hereditary disease such as Duchenne muscular dystrophy. 60% of the mutation in Duchenne muscular dystrophy are deletion of one or several exons, and this changes the reading frame, and there is a premature stop codon that will be encountered in the following exon, and again, there is no functional dystrophin protein. So we are using the CRISPR-Cas9 technology. For those who don't know CRISPR, it's clustered regulatory interspace short palindromic repeat. And this has been used by bacteria for thousands of years, millions of years, to fight virus. In 2012, this system was also shown to be able to cut the genes in plant, in animal, and in humans. And therefore, in 2020, the Nobel Prize of Chemistry was given to Doudna and Charpentier for their work. And as of today, there are 45,493 articles on CRISPR. In fact, this CRISPR-Cas9 technology uses a protein which is called Cas9 nuclease. This protein is able to induce double-strand break. It's able to cut both strands of DNA. And to indicate where this double-strand break has to occur, the system uses a guide RNA, which targets a sequence of 18 to 20 nucleotides in a DNA. This targeted sequence has to be followed by a protospacer adjacent motif, which is called a PAM, which is NJJ for the Cas9 of the Streptococcus pyogenes, which was the first Cas9 used for the CRISPR system. So whenever there is a complex form between the Cas9 nuclease, the guide RNA, and the DNA, then a double-strand break that both strands of DNA are cut at exactly 3 nucleotides from the PAM. When there is a double-strand break in the DNA, there's 2 mechanisms of repair. One is called non-homologous enjoining, and this results in micro-deletion or micro-insertion of a variable number of nucleotides. And therefore, this is not a good way of repairing the dystrophin gene, because most of the time, this would make a gene non-functional if the number of nucleotides which are deleted or inserted is not the adequate number. An alternative form of correction is the homology-directed repair, and this permits to introduce a sequence of nucleotides that may be just a few nucleotides or a complete gene at a very precise localization in the genome. However, such repair are very rare. This can be done in cells and culture, but it's not effective enough to be able to dream that this would be done in vivo to correct the dystrophin gene directly in patients. So fortunately, in 2019, there was a new development, a modification of the CRISPR technology, a modification which is called the prime editing technology. Instead of using a Cas9 nuclease, it's using a Cas9 nickase, which cuts only one of the strands of DNA. And this Cas9 nickase is fused with a reverse transcriptase. This is another enzyme which permits to synthesize a strand of DNA from an RNA template. And this system, instead of using a single guide RNA, is using a PEG RNA for prime editing guide RNA. And this is a modified single guide RNA. It still contains a sequence which targets 18 to 20 nucleotides in the genome. And then this is followed by the constant part of the single guide RNA. And then there is 2 additional sequences which are added to the single guide RNA. One is called the primer binding site. It's a sequence of about 10 nucleotides that target the strand of DNA that has been released by the cut of the DNA by the nickase. And this primer binding site is followed by another sequence, which is called the reverse transcriptase template. And this is the RNA strand that will serve as a template for the reverse transcriptase to synthesize a new strand of DNA. What is important is in this reverse transcriptase template, we may change a few nucleotides. In this slide here, the few nucleotides that are changed are in red, and this will indicate what nucleotide have to be included in the new strand of DNA, which is synthesized by the reverse transcriptase. This is the way that we can modify nucleotide in a precise gene at a precise position. Nuclei change only 1, 2, or 3 nucleotide exactly as we want. So we have been using this technology to correct in vitro nonsense mutation in the DMD gene and trying to correct the reading frame shift caused by deletion or insertion. And so this technology permits not only to modify a nucleotide, but also permits to include or remove a specific nucleotide. So the initial experiments that we did with the prime editing technology, we were able to correct about 5% of the point mutation of a DMD gene for cells in culture. And this was a bit disappointing, but we kept working on this technology and we have improved it. For example, here we have the correction of a premature stop codon in exon 59 of the DMD gene. In this case, the stop codon, the TGA sequence, will stop the synthesis of the dystrophin protein exactly at that point. It's due to a mutation. Normally, it should be a C in that position, and instead of having a C, we have a T. And therefore, to make this change this T into a C so that we have the normal codon for arginine, we first have to identify a PAM, which I remind you is an NJJ sequence, N being any nucleotide. So as you can see in the yellow square, we have identified a PAM, which is not far from the target nucleotide to be modified. And then we have a protospacer sequence, which is the 20 nucleotide that have to be targeted by the PEG RNA to be able to do the modification. So for each of the point mutation that we want to correct, we have to do that. We have to identify a PAM, which is not far. We have to identify a protospacer sequence, and then we have to synthesize a PEG RNA that will permit to introduce the mutation exactly at the site that we want. Oops. Okay, so using various modification of the prime editing technology, such as adding an additional single guide RNA that induce a cut, a nick in the other strand of DNA that is not targeted by the PEG RNA, or mutating the PAM to prevent the loss of an already introduced modification, we were able to introduce, following a single treatment of cells in culture, 36% of correction of a mutation. So eventually, after we realized that that the correction that we obtain depends on the distance between the nucleotide and the PAM that we have identified. And depending on that distance, we can obtain up to 60% correction of a point mutation in the DMD gene after a single treatment of cells in culture again. So we have published our results, and we have been able to modify several different point mutation in the DMD gene. We have published our results in 4 different articles. So the aim of the new defeatduchenne grant is to increase the frequency of corrections of point mutation. And one of my students is currently at a meeting in Banff, and she reported to me that some researchers have reported a recent chemical modification of the PEG RNA, which makes the PEG RNA more stable, because the PEG RNA is a sequence of RNA that can be degraded by the cell exonuclease. So a chemical modification of the PEG RNA increase the resistance to the exonuclease and therefore increase the frequency of prime editing. So we are looking forward to try that. And a second objective of the grant is to correct the reading frame due to deletion. And again, these experiments will be done in vitro, in cells and culture. And we want to do these experiments in vivo. And so we want to correct the reading frame by inserting or deleting one or two nucleotides or by modifying the splice acceptor as the splice donor in order to restore the normal reading frame of the dystrophin gene. So the main problem of gene therapy has been, and it's still, delivery, and delivery. So to deliver the component of the prime editing technology, we unfortunately have to use two adeno-associated virus. We could use the myoAAV, which is a very good serotype of AAV to target the muscle, but yet we have to use two AAVs, and the production of these AAV for human clinical trial is extremely expensive, and therefore gene therapy treatment are $2 to $3 million per patient. So it's nice to develop gene therapy, but we have to develop gene therapy which is affordable by the health system of Canada, and therefore we are working on the delivery of the prime editing component with the lipid nanoparticle. The lipid nanoparticle have been shown to be able to deliver messenger RNA. This has been used by Pfizer and Moderna for the COVID vaccine, and this has been very successful. We are also working on another delivery system, which is the extracellular vesicle. These extracellular vesicles are small particles of about 100 nanometer, which are produced by every cell in the human body, and therefore they are extremely abundant in the human serum. They contain 10 to the 15 extracellular vesicles per liter, and we have been recently able to introduce the Cas9 nuclease protein inside the extracellular vesicle. We're still working to introduce the Cas9 nicase fused with the reverse transcriptase. And finally, another method that has been recently developed is called virus-like particle. It permits to deliver protein instead of delivering RNA. It's a modified retrovirus. So our final aim of this type of research is, of course, to obtain approbation of Health Canada of a clinical trial of prime editing for Duchenne muscular dystrophy. And I would like to thank again the support of Duchenne muscular, Defeat Duchenne for the grant, and this will permit us to continue this research project. And what is very interesting with the modification of a nucleotide in a gene is there are about 7,000 different hereditary disease. A large percentage of these hereditary disease are due to a single nucleotide modification. So progress of the prime editing technology may eventually be a treatment not only for Duchenne muscular dystrophy, but perhaps for dozens of other hereditary disease. Thank you.